And I was very much impressed, or still am very much impressed with Jean-Marcel's skills in the different areas, not only in science, even management of the people, and also management of funds. So this is really remarkable skill that you are having all the different skills together. And Jean-Marcel has a particular interest in promoting modern breeding methods to hasten crop improvement in the developing world, helping to weave effective and interactive communities of crop researchers at both the global and regional levels, bridging the gap between basic and applied agricultural sciences. He believes in true partnership and solid capacity building to overcome some of the bottlenecks in research for development, with developing country partners as key actors and leaders in the research arena. We are very much pleased to have him with us, so please join me in welcoming Jean-Marcel once again. And Jean-Marcel is here.
Thanks a lot, Rajiv. It's really a pleasure to be here, so thanks for the invitation to this great event. Thanks also to David Burvingson for ICRISAT hosting us. And it's, of course, a privilege to now speak after Dr. Swaminathan. I would like to tell you a little bit about the story that, is a story that's closed in December last year. We had the Generation Challenge program for a decade, and I think I would like to share some of the lessons learned and conclusion from this story. So my presentation today will be about just one slide about translational biology. I would like to introduce the Generation Challenge program, talk about major achievements, a few slides on the integrated breeding platform, lessons learned and legacy, and some perspective and conclusion. So first, translational biology. I mean, this is something that is extremely important, and we mentioned this several times during the different talks over the last days. A lot of good intent, but still, I have to say, too little impact for now. If we look at translational biology, we can probably divide it into two components, a research component and a deployment component. And although we made really tremendous progress on the research side, really linking upstream research with applied component through different pipeline, the Gates Foundation project, African Orphan Crop Consortium, this event bringing people together from different fields. I have to say that still a lot needs to be done on the deployment and especially looking at sustainable adoption. We still have too much of a paternalistic approach, probably. And one of the major challenges is really to continue to keep the scientists working in developing countries excited about their work. So it's about capacity building, fund recognition, partnership, professional development, and etc. And I would say that probably if we want to have a sustainable adoption, we need to start by implementing good practices. The baseline in most of those countries is quite low. So infrastructure, data and knowledge management, quality control. So for and translational biology to be successful, it's really about change management, and the human component should not be underestimated. And I will come back on this challenge later on. So just one overview slides about the Generation Challenge program. So we have been created about 10 years ago. I'm tempted to say we are a baby of the perpetual revolution of the CGIAR. Because things are changing very, very fast. It was by that time to create challenge program to bring partnerships, raise the level of quality of the research, attract new funding, and it was clearly a time-bound effort. So we initiated our main activity in 2004, and as I said, we closed our research activities in December last year. We had a budget of about 15 to 17 million. We targeted drought-prone environment in sub-Saharan Africa, Southeast Asia, and Latin America. We were working in phase 1 on 18 CGI-augmented crops and focus in phase 2 on 9 crops. And really, our focus is on how to use genetic diversity and to provide breeders with access to new tools to boost crop improvement for greater food security in the developing world. It's important to understand that we didn't run science ourselves, but we'd rather where a broker in plant science, bringing and making funds and available to different partners to run the research. And again, one of the major challenges was really to bridge the gaps between upstream and applied science. So phase 2 was structured around 7 research initiatives. We had one on cassava, one on legumes, one on maize, one on rice, one on sorghum, one on wheat, and the last one was on comparative genomics. And the trends of all those different research initiatives that were focusing on few target countries in developing world was really to go through this classic path about identifying suitable germplasm, using genomic resources to identify genes and alleles, and then through the development of a platform, how to bring this information into adapted genetic background to end up with improved germplasm. Our initiative overall was quite a big one. We had about 170 millions over these 10-year periods, and you have the different main donors are listed here, and without any doubt, the European Commission, DFID, the World Bank, and we had more recently also the Bill and Melinda Gates Foundation coming on board. They've been really the strong supporter of the program. Now, I would like to come on some key achievement, and I will not focus necessarily on research output, but perhaps a bit what we learn also from a people perspective. And I would like to start with what I call really true partnership, because I think that has been without any doubt one of the major achievements of the Generation Challenge program. And when I talk about true partnership, I think about tangible partnership where through different indicators, you can really see that people are engaged. And indicators include money allocation to the different partners, significant in-kind contribution from them, open exchange of results and in information, and really to attract partners, not necessarily because you are a funding agency, but really because they want to be part of a big network and give them some visibility. This true partnership must build on trust and goodwill. And one also important criteria of success is that even after the GCP, most of the networks that we developed and implemented continue to work together. What is also fundamental to be successful is that you need to have this dynamic in the responsibility across your different partners. And at the beginning of the program, leaders were coming from universities, were coming from CGI centers, and over the time, they became mentors, and thanks to good knowledge application and transfer, the trainee who came from the developing countries, they became the doer and the leaders. And again, this dynamic is absolutely fundamental for sustainable adoption. We end up in phase 2 to have more than half of the PI coming from developing countries, and again, half of the money were going directly to them. I would like to underline, however, since this has been something that is too often underestimated, that it takes time and resources to develop and nurture a true partnership. Linking upstream with applied science, here I would like to take this example about finding a gene for aluminum tolerance in sorghum. It started through competitive grants that a team at Cornell, led by Leon Caution in collaboration with Brazilian scientists, won, and they started by evaluating collection of sorghum under hydroponics, and they found that the major gene was responsible for quite a number of or a large percentage of the phenotypic variants, and they were able to clone these genes. Through a second project, they were able to look at Embrapa germplasm, be able to screen different collections that they had, identify the favorable alleles at these genes, and start to improve the germplasm from Brazil. Looking and build it on this success, we commissioned work for the last 5 years of the program, and then we supported the transfer of those favorable alleles from Brazil to Africa, and we end up by converting some African germplasm with this aluminum tolerance gene, and now we have improved germplasm in in African soil at both Kenya and Niger. So I think this is quite a unique and nice example of within 10 years, you started with a collaboration at the university, prestigious university in the US, and you end up with tangible product, in Africa with a stop in Brazil. And Leon continued to be engaged in this project and, continue to mentor and help those people, and the same for the Brazilian team. So this is really, I think, something that we are very proud of. Here's the slides of different major research outputs. Again, don't want necessarily to go into too much detail here. We have a product catalog that is available, on our website, but briefly, we generated quite a number of genetic resources. Genomic resources in particular, we are very proud that when we initiated 10 years ago, we had a lot of called orphan crops, in our basket. And thanks, among others, to Rajiv Varshney and the effort that we did in our subprogram 2, we developed resources for quite a number of legumes, cassava, and others. And now we have, as I will show you later on, markers, SNP markers that are available for most of those crops. We identified informative markers, gene and QTL. We developed quite a number of germplasm and worked also on bioinformatic tools. I think one element that was also quite critical was the capacity building components, and I will come back on this in one of my upcoming slides. So again, quite a number of substantial products have been generated, and they are available as indicated in this website. Another element which is quite important is that how you measure the quality of the science and sustainable adoption of your product, this one component, but also peer-reviewed publication. This is a summary of the number of publications that have been published by members of the network. This is about 500 publications overall. Still, we are a bit short and need to update the publication for 2013 and 2014. And as you can see, we had also quite a number of papers published in quite prestigious journals. So I think that's another important element when you look back and try to have a good evaluation of the quality of the research conducted. Capacity building, I think that again, you cannot imagine transfer of technology in a sustainable way without people on the ground being trained. And we had different activities, more classical approach, through formal postgraduate training programs, different workshops, and the development of communities of practice. And this in different universities, in WACI, in ACI, and other partner in Africa and in Asia. But perhaps what I would like to underline here, that we also thought a little bit about out of the box and try to develop the concept of capacity building a la carte, where really within a project, we were allocating a certain amount of money to people and asking them basically, what do you need? You identified, where is the gap, what kind of technology you need to access to be able to run your research successfully. We also developed on molecular breeding a multi-year courses, so it's not just a one-shot course. People were coming every year and they needed to bring, the output of their research to be accepted in the next course, so really showing that they were using the tools and putting in practice what they learned in the previous course. Very important also, don't train just one piece of the equation, one element of the equation. And we have scientists, we have technicians, and also station managers that needs to be updated. And we develop also special courses for those people. And I think through the platform, we try, and again, I will talk about the platform in a few minutes, but also to try to a bit shift this paradigm about the training, that instead of organizing a course on the topic, and again, this is not opposite to academic training, which is a must-have, but trying to shift to probably something that we can talk and call learning as you go. So really allowing the user that will find, some bottleneck or some issues, then to immediately find the resources to overpass these bottlenecks. So responding to the demand when people are facing, some challenge in terms of knowledge. It's like your kids today. They don't read the manual of their iPhone. They play with it. If there is something that they cannot do, they chat to WhatsApp or whatever. They send a question, somebody give them the answer, and they go to the next step. So a bit along those lines. Now I would like to pass to integrated breeding platform, because this is what will survive the Generation Challenge program, and we believe this is a very important means also for putting in practice a lot of what the technology and the results that will be generated through the genomics. So you can access the platform at this site. It builds on really tools for running your breeding program on a day-to-day basis, some breeding services, and I will come back on this one, a training component, and a community resource. And we believe that again, this will be something that is really needed for people to be able to access the new technology. Overall, the objective is really to improve the efficiency of plant breeding program in developing countries by enabling them to access modern breeding technologies, material, and related information in a centralized and integrated and practical manner. But also, as I just said, we believe this is a platform to facilitate sustainable adoption and implementation of translational research output to serve plant breeding's need. And I will illustrate this through an example. In the core of the integrated breeding platform, this is a breeding management system, and I just have one slide to illustrate a bit what the tool can achieve for you. Again, don't want to spend too much time on this, but you have all different tools and functionalities to basically run your breeding activities. You can have your germplasm running your nursery in trials, capture your data electronically. You have a set of different statistical analysis, and of course, on the top of this, you have also access to marker to run molecular breeding selections and also more recently genome-wide selection. This is not a research platform. This is really a place where breeders will find all the tools and resources that they need to do there and run their breeding program in a routine way. Talking a little bit about what I was referring to, a platform to make outputs of the research available to users can be illustrated through the breeding services component. So the platform is more than a tool. It's really offering different services, genotyping services, so the user can go there. There are different service providers. Some of them are in the room. LGC is one of them, where for the dozen of crops that are there, you can find and have access to 1,000 to 2,000 SNPs at a preferential price, so people can directly order that genotyping work through those different service providers. We have access to climate data through a company that's called Aware, that's really a professional service to bring GIS information to breeders, phenotyping, and access to other software. But here again, I would like to emphasize, a resource for crop information and genomics, and here you can also access diagnostic markers. And if you click on diagnostic markers, you will find a list of markers that have been generated, through, at this stage, this is a work that has been done through the Generation Challenge program, but we want to expand this list. This is the marker that Gary referred to as the must-have, the one that you can do and use it in a predictive way. And if you go on rice in sorghum, you find now this famous aluminum tolerance gene that is coming out of this nice Cornell, Brazil, African story that I was referring. And if you click on this, you will have some general information about the value of the genes. Here you find the information about the markers that are available, germplasm that have the resistance, some general information, and clicking here, it's not implemented now, but you will be able basically to order directly the markers to select these genes. So this is a typical example, again, of what is coming out from this 10-year research that I was referring to, can be today directly used by breeders in their breeding program. We have been lucky to have some supports to go for a second phase. This is a project that has been supported during phase 1 and continue to be supported in phase 2 by the Bill and Melinda Gates Foundation. And I'm very proud to say that David Burvington was the first project officer at the foundation who worked with us to develop this concept, and now Gary is taking on and is leading with us the phase 2. We have also some resources coming from the Generation Challenge program, and really after 5 years of phase 1 focusing on development, we are now focusing on deployment. And you can believe me, the first phase was quite challenging, but looking back, this was the easy part of the equation. The deployment part, boy, I mean, this is really something, you know, how you change people's behavior, and even if it's free, and even if you work with them, and even if they see the benefits, this remains a huge challenge. So we are going to work very hard and consult with people and together try to really have the platform adopted. So no doubt that behavior changes is the major challenge. And when we want to talk about sustainable adoption, again, we need to start very simple. Good breeding practices, how you manage your data. So we have different baseline depending on the breeding program format and shape. And again, you know, we'll have to think about specific deployment plan for the different partners that we are going to work with. And very important, one size doesn't fit all. So be flexible. Also fundamental when you have to have a sustainable deployment, you know, you need to have local support. And we developed some regional hubs. In red, you have the hubs that are already implemented. We have 4 in Africa. We have 3 in Asia. The one in black are the one to come. And here this is where we have our HQ, you know, hosted by CIMIT in Mexico. And again, I mean, the users will therefore be able to locally have support from a technical point of view if they face any issue in the use and application of the breeding management system. But we have also a vision that go a bit beyond the current IBP, and I would like quickly to show you what this vision is. So if we put in the middle of the IBP the breeding management system, which is really the core analytical pipeline, as I said, we have crop resources that are available, services, breeding services, we have capacity building, we have space for a social network, and these form the current integrated breeding platform. But we are now looking for bringing a station management system, and we are working with a company in Canada called Agricon. We have a system that has been already implemented in Africa that might be relevant. Breeding management system at IRI, they did a tremendous job to put in place, you know, a system that will drive, you know, the breeding activity through a pipeline path, and we are looking and working with them to have this now adopted and implemented in the BMS, if suitable for our different users. We have access, as I indicated, to GIS management system, and we plan, the idea is also that we bring on board the finance management system. So at the end, you know, for a small program, they cannot afford to have different management system, okay? And if we are able to achieve this in the midterm, you know, few years, then hopefully, you know, this platform can really become their integrated breeding management system, and the links are obvious. You know, you can think about, you know, a project, you developed the outline of this project, you have the link with your finance people, then will implement their experiment, you know, directly they can send some information to the station manager in terms of the plot, what they would like to use. They can use some GIS resource, and of course, the cost for doing those different activities, you know, can also be linked with the finance management system. So this is a bit, you know, the more global vision of where we try to go. I will have a couple of slides now from lessons learned and conclusion from the GCP experience. Saying that, you know, although we believe that it has been quite a successful experience, and we had an external review that's available on our website that I believe, you know, confirmed that most of the objectives have been achieved in a suitable manner. It's still, you know, difficult to really quantify impact, because although we have quite a number of output and product, they still need to be used. And it's only in a few years about the use of those products that we will really be able to validate the success of the program. Quality of science, I think, as I said, has been quite good, supported by quite a number of publications. A broad range of products have been generated and are available. You know, it's not only somewhere, it is, you click on our website, you will have the contact person, and some of those, as I said, have been transferred to the platform to be used. So we went a bit down just the output and really focus about making the right shape and the right mean for making those products available to the different users. The network will continue to operate, as I indicated, when we will stop. And some people already missed the GCP, which I think this is quite a good criteria of success. Major achievements have probably been around, really establishing a true and dynamic partnership based on trust and evolution of responsibility, and we create this kind of GCP spirit. And I will show you an example of this GCP spirit later on. It's also about a cultural change, how to run research for development projects from research and a management perspective, and really to put at the end of the day partners from developing country first, and allowing them to become leaders and access new technologies. The combination of competitive and commission project has been absolutely key to our success. Too often we believe that we know people working in the same field as us, and we try to invite them to work together, while competitive project definitely broaden the scope, bring new people on board that you had no idea they were doing or conducting research in the same area, and also creating new partnerships. This model of a broker in plant science, I think, is really a very attractive model. Of course, it cannot stand alone. It needs to build on strong institutional activities. But perhaps it's time from some of the institutions, I can imagine perhaps in ICRISAT, you can allocate a portion of your budget perhaps to this kind of model, where you have a clear focused research objective, time-bound, and allocate this money and create through collaboration with different partners this kind of dynamic that we created through the Generation Challenge program. And hopefully a lot of lessons that we drawn from the Challenge program can be applied to other operational models, including the CRPs. The legacy, as I said, is the IBP, and we'll go ahead, continue to contribute and promote breeding evolution. I'm tempted to say revolution. I mean, we talked a lot about, how we bring the revolution, or the green revolution, or the blue revolution, or the new revolution to Africa. Perhaps a very good entry point is to, again, bring good breeding practices there. What kind of technology, what kind of infrastructure, what kind of data management you can bring, so that you can really build on this revolution. The idea there is, again, the one-stop approach. So, to centralize those days, you don't have the time to go at different sites to access different technology or tools. And again, quite important for making the links between upstream research and providing some of the products through the platform at the disposal of the different breeder. As I indicated, the breeding, the capacity building, the support as you go is also quite important. And again, no time to talk about this, but we are talking here about an innovative business model. Some of the people will pay to access some element of the platform, and we try to make it, after 5 years, sustainable, at least from the development and the maintenance of the platform per se. The BMS is building a very good momentum with increasing demand, but here again, this is only when we'll look at adoption, who is really using and adopting it, that we can measure the impact. And we have already now, some centers. I mean, CIMIT, ICRISAT, are really engaged in implementing and deploying the BMS in their own institution, plus other partners from national programs. So we have a momentum here, but believe me, also quite a number of challenges in front of us. This is a list of the different people that work, and were involved in the Generation Challenge program adventure over the last 10 years, quite a long list. And this is the picture of the last annual meeting of the Generation Challenge program. And here, if you look at this, this is a good illustration of the Generation Challenge program spirit. Sorry about this, Rajiv, but here you have Leon Koshian. Leon is the PI of this famous aluminum tolerance project, and just making a clin d'oeil, as we'll say in French, to Rajiv. Now we have just 3 last slides to come back again to the topic of this conference, really about the genomics and its application. I mean, we heard a lot of very good story, and this is a lot of promises there, and we are really going the right direction. But I think it's also very important to keep in mind that the challenge is big. I mean, this is, for example, epistatic effect in maize for female flowering on a biparental population that has been measured on 9 different environments, different location, different water regime. And you have the different chromosome represented here, and there you have the epistatic effect. Under abiotic stress, which is the target stress, of most breeding activities in developing countries, additive effects go down. Absolutely no doubt about this. Epistasis is more and more important. Epigenetics is also more important. And you can see that those plants, they are damn smart, Depending on the environment, depending on of where they are and how they grow, they put in place different mechanisms, different genetic networks to respond to the environment. And we need to keep in mind that the response is not the one we want. The response for them is to survive, so to produce at least 1 grain. So it's not only to understand how they behave, it's also to understand how they behave and then ensure that, they will produce more grain under this unfavorable condition. Good news is that we have some regions that definitely continue to be driver, probably cluster or 1 gene for transcriptional factor and other major genes. But again, I think this illustrates very well the point that it's a complicated system. And also I would like to come back perhaps during some of those presentations and not talking really about genetic studies. I mean, I think this is quite clear, how progress are made and the way we go to take advantage of genome-wide selection, other approach. But really, when we talk about resequencing all those genomes and etc., some of the talks, give a bit of flavor of the miracle approach. And what I mean by the miracle approach is that we hear, and we are going to resequence all this germplasm, and we are going to find the alleles, and this information will be used by breeders. Well, it will be nice to know a little bit more about the information that will be used by breeders. And I think that's a real challenge for the entire community. And probably my definition of translational biology is really to increase the probability that the miracle occurs. I would like to finish, however, with a very good, note. I mean, without any doubt, the plant genome is unveiled today. I mean, this is really a major achievement, and ta-da! That's what we get. And now, of course, in everybody's minds, this is okay. Holy, this is really complicated. Now, what are we going to do with this? And I think this is really a great question. So I have an answer for you, and I think the mission for this community is really clearly defined, and ta-da! This is what we are trying to produce over the coming years. Thank you very much.
As usual, fantastic. You can take your seat. So once again, we are very much grateful to Jean-Marcel for this great presentation. So please join me once again.